Hi, everybody. Thanks for joining us this afternoon. As Azure had mentioned, I'm Tracey Dalton, Director of the Rhode Island Sea Grant Program, and I'm so pleased to be here today. And we're thrilled to have you all joining us for this Bay SAMP webinar. So today, as Azure had mentioned, we're fortunate to hear from two scientists who've been doing some really exciting research here in Rhode Island and Narragansett Bay and Rhode Island's coastal waters. And as Azure had mentioned, our speakers are going to give some presentations. They're going to have give maybe a 15-20 minute presentation. We ask that you hold your questions until after the presentations. You can certainly put them in the chat now, but we'll answer them after the two presentations. And I will go ahead and read them to our speakers. So let me, without further ado, let me introduce our speakers now. I'll introduce both and then we'll get started. So our first presenter is Chris Kincaid. Chris is a professor at the URI Graduate School of Oceanography. Chris studies the physical environment of Rhode Island coastal waters, the how, why, and where of circulation, transport, and flushing of water in Rhode Island Sound, and within all the interconnected passages, rivers, embayments, and coves of Narragansett Bay. So today he's going to present an update on results from recent advances in both observations and modeling of local ocean physics with implications for bay ecology. Our second presenter is Robinson Fulweiler, Wally Fulweiler. She's a professor in the Department of Earth and Environment and Biology at Boston University. She's an ecosystems ecologist and biogeochemist whose research is focused on understanding human impacts on marine systems. She heads the Coastal Ecology and Biogeochemistry Lab at Boston University, where they study energy flow and cycling of nutrients, carbon, and oxygen in different environments. Today, she's going to be talking to us about oysters and some research that her lab has been doing on understanding how oysters alter coastal nitrogen cycling. So some exciting talks ahead, and we'll start with Chris. Are you ready to go, Chris?
I think I am.
All right, take it away.
So should I share my screen?
Sure.
So can you see the PowerPoint?
Yes, you might want to do the
full
start. Yep.
Okay, can you see that okay? All right, so I'm going to talk a little bit about the work I've been doing with looking at circulation in Narragansett Bay. It's kind of fun to be on this with Wally because Wally and I have worked together for quite a few years. Some of the data I'm going to talk about today dates back to the last century, and Wally was there to help collect it.
Thanks, Chris.
Sorry, Wally. Yeah, so these are the things that were in the announcements, looking at bringing perspectives on Narragansett Bay, some latest research, particularly pertaining to aquaculture. So I'm just going to start off with the whole thing that I'm kind of a coastal plumber. I look at how water moves and flushes in Rhode Island waters from Rhode Island Sound into Narragansett Bay, shown here. And there's a number of goals associated with circulation and everything we do at the Bay. Certainly one of them is balancing the health of the ecosystem with economic health drivers of a good economy. What I'm showing you on the right side here is data from the Insomniacs cruises, which were done many years ago. This is from 2008, but it's showing the bottom dissolved oxygen levels in the Bay. And every place there's a little dot there is where people in boats went out overnight and worked from like sundown to sunup, collecting data on oxygen levels. And it was really sort of a groundbreaking thing because it showed us sort of in great spatial detail what the dissolved oxygen looked like in Narragansett Bay. And the red areas are areas that have low dissolved oxygen. And so in terms of a management tool, trying to manage things like healthy ecosystems, you could say that you want a tool that would predict all of the Narragansett Bay parameters, every single one of them, physics, chemistry, biology, everything, at all points in the Bay for all times and for all conditions, past, present, and future. That would be sort of the dream. And one of the hopes for that is are computer-based models of ecosystems. And the point I want to make is that it's sort of a foundational thing. So if you think about a management tool that can do everything that I just mentioned, and the pinnacle of the diamond is sort of the ecosystem or the biological processes. At the foundation, one of the foundations is the physics, the physical, the transport, the flushing, and the mixing. And that's what I'm going to talk about today a little bit. And if we're looking for motivation, certainly the fish kill of 2003 is one of the reasons we want one of these management tools to look at water quality, nitrogen dynamics, blooms, low oxygen events. The other one that's sort of certainly coming along is harmful algal blooms and things like rust tides. This is a clip from an article about rust tide problems in South Korea. So I'm just going to give a quick overview of the three-legged stool for coastal ocean physics. Wally, I have to put this in when I give talks, right? This is like your favorite diagram right here. Yeah. Okay. So the three-legged stool is data, observations of physics. 2 is lab models. The lab I grew up in was really big into building physical models of the systems you're looking at, and certainly computer models we all know of. So I'm just going to show you some snippets of data that we've collected. We basically, since the 90s, we've collected observational physics data in every nook and cranny of Narragansett Bay and out into Rhode Island Sound as well. So I'm going to just show you some examples of how we build better models using these 3 legs of the stool for the Providence River in a particular area called the Edgewood Shoals in the Providence River. So that's shown over here on the left. And here's a map of the Edgewood Shoals, and each one of these little vectors here is a flow vector. And that's from, we had 25 tilt current meters out, these little simple meters that sit on the ocean floor. And we've had them out in 2009 and 2010. And unfortunately, in 2010, we caught the Great Flood of 2010 that occurred March 29th. And I'm going to hit go on this movie and see if it goes. Okay, and so what you're seeing here, this is the detided flow in the near bottom water on Edgewood Shoals. And I'm kind of showing the shipping channel in blue here and the shallow shoals of Edgewood over here, which if you look over on the left here, that's one of the chronic areas of low oxygen, that Edgewood Shoals thing. And so this just shows what you'll see very prominently in all the data that we collect on Edgewood Shoals is that there's a really prominent recirculation gyre of the bottom water that very slowly spins. If I go to the next slide, here's the currents during the flood. The flood hit on March 29th. So if you watch the date here, you'll click along, you'll see it come into the 29th. You'll see the flow vectors push to the northeast. That's when the storm comes through. And then they swing around to the south, southwest, and that's when the peak of the flood goes on. And one thing that's remarkable is that this is one of the largest outflow events that's occurred in Narragansett Bay, and it does nothing to the shoal. It does not flush it. So this is just like a summary of what that flow looks like. The gyre is amazingly stable. The Great Flood was not even able to break it. And models, if you're going to build a model that's believable, it has to represent things like this. And we find these gyres in every little subsystem, all the areas of Narragansett Bay that have the lowest oxygen levels, Bristol Harbor, Seekonk River, Greenwich Bay, Inner Greenwich Bay, they all exhibit these really stable flow gyres. The other data I want to show you is just show you how severe this is and how hard this is for models to get. So if we go out periodically. Wally is not on this boat. I could never get her to do one of these. These are full tide cycle surveys. Often you go at night so that you don't have bad weather. And you're driving for 16 hours back and forth out there. It's cutting the grass. And these give you remarkably detailed images. This is the data from one of the surveys at Edgewood Shoals. So you're looking northward into the cross section. So it's depth in the vertical, it's east on the right side, and it's west over here on the left side. This is the shallow shoal. This is the deep channel. And the reds are water that's going away from you. Blues are coming at you. And the thing that's remarkable is that, or repeatable, is that you see this blue outflow coming right at the edge of the shoal. You have this deep inflow over here, and you have these gyres and spinners up here on the shelf. And if we take the detided flow, the residual flow it's called, that moves stuff around, it's a hydrodynamic disconnect between what's happening in the channel and what happens over here on the shoals. It's an incredibly sharp boundary. So that's data. I could show you many, many more instances of it, but I'm not. The other leg of the stool, one other is lab models. So when I happened to be in Australia on a research sabbatical, I stole a lab setup there that was perfect for the Edgewood Shoals. This is it here looking down from the top. It was a pre-built, it was all set to go. It had a shipping channel and it had a shoal right off next to it. So the deep water is here and the shallow water is here. And we, in a lab model, you can do all kinds of cool things. So we had river flow coming in from the north and we have tides on and off down here. So I'm just going to show the movie. The reason you do lab models, and one thing, I don't have a date thing running on here, but every time you see the stuff shift down and then shift back up, that's a 12-hour period. And so the lab models are brilliant because they don't have grid resolution issues. They're real fluids. And so one thing this shows remarkably well is how a setup like this flushes and interacts with the channel. And so this helps us to understand the physics and the small scale eddies that control the exchange between the gyre and the channel.
Chris, I'm sorry to interrupt you, but we've had a request if you could try to put your PowerPoint in presenter mode, just so it would show up bigger on some folks' screen.
Yeah, unfortunately I can't, I'm not seeing it in my, I tried to do that earlier. Okay, let me see if I can
be able to do it on slideshow.
Presenter view. Does that work?
No. Let's see. You want, yeah, get out of that. Slideshow, the next one. Perfect. Yep. Now you have to scroll through. Thank you.
It's okay. Okay. So the next thing, the next layer of the stool is computer models. And I just I'll just finish on that. So we've built multiple versions of this model called ROMS, Regional Ocean Modeling System. And we've applied that to Edgewood Shoals also. We've tried to train it. And the way a model works is that, so now I can't see what's on the other side over here. So how do I compress this thing? I'm not gonna be able to do it. A model basically breaks the domain up into a set of cells, and it solves these complex mathematical equations by taking really crazy math and breaking it down into simple geometric expressions between neighboring nodes. So the heart and soul of a computer model is the spacing of the grid nodes that it's using. And it's very much like a camera with pixel resolution. So if you have a picture that's pixelated, that has a small number of pixels, like the only phone I'm allowed, like I have a phone from the 1920s. So I don't take very good pictures. And so that relates in the model. So this is a computer model using ROMS they call it ROMS 1 from 2005, that shows flow on the Edgewood Shoals. And what you can see with this particular course model is you get a blurry picture, and it predicts perfect flushing of the Edgewood Shoals. And through time, we've added resolution. We beef up the number of nodes, and that allows us to actually represent fairly well the gyre and the flushing. And we're doing this not just at Edgewood, but in every particular place. And then into the computer models, we can put things like dyes and floats to track what's going on. This shows just, in this particular one, we have put the nitrogen output from the Fields Point Wastewater Treatment Facility, and we can track where this goes over time. What's kind of cool about this is that we can individually track every single source of fresh water, every river, and every wastewater treatment facility. So we have 25 different chemical dyes operating in these models. So it's almost a bit like forensic oceanography, but it's never good enough. The problem is I can show you a model that matches data perfectly in one area and it's total crap in another area, right? And so, these are my models. I'm not throwing anyone else's models under the bus. My computer models from 10 years ago are crap. So, but anyway, so this ROMS 2 that I just mentioned, it does really well at representing the gyre, but it misses in a lot of key areas. It can't get the exchange at Seekonk. It misses badly at Bullocks. It misses certain key things in Bristol and Greenwich. And so what we did was we put out current meters right in the worst area, like Bullocks Reach. We said, what's going on there? And every time we put in more current meters, we can start to get a sense of what's going on. And we really could see, I'm gonna show you a movie. This just shows for this new version of model that we've just finished in 2021, it's able to recreate the flows extremely well between the channel and the shoals down at Bullocks. And so what you're seeing is releases of floats into the model. the blue is into the bottom water, and the green is mid-water, and the red is surface water. And so these are the kind of things that we could put around aquaculture farms, and we could predict where the flow comes from that enters the farm and where it goes. How am I doing on time?
You're good.
All right, I'm going to show you one application. I can stop at absolutely any time because I'm only here because I want to hear Wally speak. So I'm going to just show you one application of this model. So what we did, we had a version of physics that we thought was pretty darn good. And we made a simple biomodel, NPZD, nitrogen, phytoplankton, zooplankton, and detritus. Wally's shaking her head, yes, so that's good. So what we did, we were really focused on nitrogen sources from the north and looking at phytoplankton blooms. And what we found from the case I'm going to show you is that the nitrogen is largely coming from the north, but it's also coming from all these other areas. But the blooms seem to grow in the mid bay, and then they move northward. And so you get phytoplankton riding currents up into the Providence River and just going berserk. So here's a movie showing, this is from ROMS 2. This is the model that is okay, but not great. And it shows the red is nitrogen coming from the northern rivers and wastewater treatment facilities. And you can see it coming in from all these other areas. And then this over here is a phytoplankton bloom that's happening. This is from 2010, June of 2010. And you can see every place is red is where this phytoplankton is starting to grow. And what's kind of cool about this is that this is one of these ones that starts to evolve in the lower Providence River and Ohio Ledge. And then it very rapidly moves northward, as far up into the Sequoia River even. So this is just an application that we did. And so one thing that we did was trying to test the effect of reducing wastewater treatment facility inputs. So we ran this with old inputs, and then we dropped it down the levels. And so I think what you can see in this is I'm keeping track of phytoplankton bloom amount at 6 different places. And those numbers correspond to the numbers here. And then I'm just showing different cases for when we drop from old releases to 5 milligrams to 3. And you can see improvement in some areas, not in others. And you can see big improvements in some areas and not others. Okay, so that's sort of a overview of stuff we've done. The stuff we're working on now is trying to improve. This is the stuff I'm doing with Wally, is to improve estimates for the nitrogen budget. What comes in through the mouth, that's sort of a forgotten piece. And we're also starting to look at rust tides. And so the last little bit here is we have awesome data from the Nixon lab from the turn of the century. Sorry again, Wally, where they went out and they did crazy cruises along this yellow line from the bay all the way out into Rhode Island Sound. And they measured, this is now a cross section going from the bay on the left to Rhode Island Sound. What they found were high nitrogen DIN levels out in Rhode Island Sound. And that's awesome. That's super cool. But does that get into the bay? What about, is that intrude? And so one of the things we did back then, I have this great picture, Wally, of you, this picture. When we had an instrument underneath the Newport Bridge, right up against the bridge pile. And I have this great picture of Wally as a grad student, like standing on the boat, trying to climb up the bridge pile on the cable, basically, that we had running up. And we had instruments out off GSO dock and in the East Passage, and they showed like the West Passage, if you want to know where the water goes out, it's out all the time off the GSO dock, always, never goes north, on an average sense. And we see this beautiful intrusion in the East Passage. But the problem is you can't, from one point in a cross section, you can't calculate the flux. So what we did in 2018 and 2019 is we put 3 current meters across at Castle Hill. And this is a cross-sectional view. Every place you see red colors are intrusions of water into the bay. And every place you see blue is weak, 0 intrusion entering the bay. And this is across, like Castle Hill is on the right. And what's that cove over there? Macro Cove, I think it is, over there on the west side, northwest side of this cross-section. So what you see is these big intrusions coming in. And the thing that's kind of cool is that they they're very, very well behaved. They're predictable. Spring tides are relatively low intrusions. Neap tides can be massive. And when you get nice winds combining with neaps, you can get really, truly huge things. So just a quick point. If you think about the volume of these things, here's the amount of water coming over the Blackstone Dam here. All the wastewater treatment facilities together are like this fireboat. And the amount of water coming in these intrusions is like what comes over Niagara Falls. That's the average. But when these intrusions are running, they're like 2 times what comes over Niagara Falls. So these things are truly large, and we don't know what's in them. So one thing that Wally and I did was we tried to say, okay, if we have this great flux coming that we've constrained with new measurements, we have the data that she collected with Scott Nixon and others, and we can combine this to calculate nutrient fluxes. And so what we do is we estimate for 2018, when we had the instruments out, these are the moles of nitrogen coming in through the rivers, okay? And I use a minimum and a maximum estimate for nitrogen. This is the nitrogen contribution from the wastewater treatment facilities from the pre-fish kill days and the current days. And this is what's coming in nitrogen in the deep water through this deep intrusion. And what's kind of cool about it is that if you remember from that movie, the deep red cores, they're all below the photic zone. So these things are coming in and they're riding their way north deep. And so one thing that we're doing in our new version of ROMS 3, and I swear this is my last slide, is that we're starting to track this nitrogen source. And what's so cool about this new model, I don't know if you can tell I'm excited about it, but I'm excited about it, is that this is tracking the nitrogen in that deep intrusion is the red colors, and it's all the nitrogen in the bottom 2 meters of the water. So what you can see is that we have enough resolution, pixelation pixels in all the channels of the bay that you can actually see these stringers that are coming off. It gets the shipping channel that goes to Quonset. We can really track where this deep nitrogen goes. This is where it looks like later in the summer. And when you keep track of this thing, now that we're doing these biomodels again with this offshore source, it's important. So anyways, I think that might be so that's an overview of what we've been doing over the years to improve the physics parts of our models. We're pretty confident now that we're doing a much better job in the channels, coves, and embayments, the man-made channels and the natural channels. I think we know a lot more about what nitrogen comes in through the mouth. Well, at least we know what the water's doing. We still don't have great measurements of the nitrogen concentrations in that water. And we're starting to look at HABs and rust tides. So maybe I'll stop there.
Great, thank you, Chris. All right, so we will move on to our second presenter, Wally Fulweiler. And again, as you have questions, please put them in the chat and we'll pose them at the end of the presentations. So Wally, Chris, why don't you take yours down so that she can put her presentation up?
I don't know.
Enough physics. Stop sharing, Chris.
Stop sharing. Here we go.
And you're making me feel very old. I recently had a graduate student ask me if it was okay to cite a paper from 2000. And I was like, yes, that's totally fine. Yeah. And she was like, it's not too old. Said, no.
I didn't mention that you babysat my children. I didn't mention that.
I've known you for a long time. All right, let's see if everyone can see this. Can you see a beautiful oyster? Fantastic. So thank you so much for giving me the opportunity to talk today about oysters and nitrogen, and I'll throw in a little bit of other nutrients here and there. I'm a biogeochemist and an ecologist, and what draws me to oysters is just their amazing capacity to filter water and all of the other sort of services that they provide. And I'm going to admit my bias right now and say that I think that these ecosystem engineers can help us sustain water quality and also build coastal communities. And so I'll sort of like hint and talk a little bit about that today. I also want to make sure that I acknowledge my oyster team, Nick Ray, who's currently a postdoc at Cornell, Alia Ahaj, who's finishing up her PhD here, and Gretchen McCarthy, who was an undergraduate and is now a PhD student in New Zealand. So just in case I ever say I, please know that this is a big group effort. These are very intense measurements and lots of thought from lots of people have gone into this. Okay. So I probably don't need to tell you, but I will anyway, that we humans have done some pretty intense, horrible things to coastal ecosystems. And in part, that's because we're a coastal civilization, right? We live on the coast, we rely on it for everything from our economy to food to culture and recreation, right? And so in part, because of that intense sort of interaction, many of the habitats we've destroyed. And one of the habitats that have been sort of most intensely destroyed have been oyster reefs. In fact, recent estimates put globally that we've lost about 85% of oyster reefs. This is done through habitat destruction, through overharvest, and through disease. And I always like to show these photos because I think they're such an intense reminder of the type of harvest we've done in coastal systems. So these are all oyster shells and from various places along the East Coast here, just showing you how many oysters we've harvested, the amount of biomass we've taken out. And you don't need to be a scientist or know really anything about anything to know that removing that amount of biomass or biology out of a system is going to have a profound impact. And so my group is really interested in trying to quantify that and then trying to quantify what happens when we begin to restore these ecosystems. One of the things I'm going to sort of just keep talking about and referring to is this idea of ecosystem services. And it's kind of a squidgy term that can sometimes be confusing, but the key thing is just to remember that if we think of oysters not only as these amazing filter feeders, right, they also provide these other benefits, right, that are harder to put value on. So they do things like provide habitat for a range of other important species. They can protect us from storm surge. They can decrease sediment resuspension, and they can clean up the water. My group is primarily interested in this latter part, this idea of what oysters can do to water quality and how they alter nutrient and carbon sequestration. Recent estimates put sort of these non-current economic value benefits that oysters do somewhere between $5,000 and $99,000 per hectare per year. So they are quite substantial. Okay. And then I always really like to show this figure when I talk about oysters because it just really nicely illustrates the power of these organisms as filtering nutrients, as they're able to filter nutrients. So here is the tank full of oysters, and you can see how clear the water is. And here is this sort of same water, but with no oysters, and it's super cloudy. And that's because oysters, one adult oyster can filter up to 50 gallons of water per day. And I like to try to tell people, like, imagine if you had to drink that much milk, right? I mean, it's an amazing amount of liquid that they're passing through their body. And when they do that, it means that they're really being able to filter and clean the water for us. Now, this is a super busy slide, so let me just try to focus you in. If you just follow my arrow, which you can hopefully see, I want you to look up here at this sort of greenish blob right up here. And I want you just to note that there was some historic estimates that pre-1870, the oysters in Chesapeake Bay could filter the entirety of the Chesapeake Bay in under 3 days. And then by the mid-1980s, it took 245 days for those oysters to do that filtration because we decreased the population so much. And that begins to matter when we think about what that does to carbon sequestration, to nutrient filtration, to nitrogen removal, because it means that without those oysters doing that process, then all of that material is staying in the system, and that profoundly changes how the system works. We can look at this down here if we look at the amount now of carbon sort of assimilated into the oysters, and the units don't totally matter. What I really just want to hit on is, again, in the pre-1870s, they were basically assimilating almost like 32.2 grams times 10 to the 8th grade, doesn't really matter, grams of carbon per day. And yet by the mid 80s, it was only 0.3, right? So the magnitude switch is really what I care about. And then in a more recent estimate, they went and looked at a whole bunch of coastal ecosystems and said, how much has the water, the amount of water being filtered, like the residence time of that water, how much has that changed over time? And what I want you to know here is just that across a wide range of these systems, because we've removed oysters, we've changed the amount, we've basically change the amount of the filtration capacity of that system. And so you can see it's decreased in all of these areas by substantial amounts, except for one in Florida, and we'll come back to that at the end. Now, there's good news, I think, and the good news is that we're beginning to return oysters back to coastal systems. We're doing this in a variety of ways. In one way, we're doing this through restoration. So in this figure here, if you focus on the squares, that's showing you restoration efforts for oysters. The circles are for coral reefs. But we're doing this in the US, in Europe, and in Asia. We're bringing oysters back through restoration. And then also, of course, through aquaculture. So a variety of countries are doing oyster aquaculture, with China in the lead. And then, of course, a variety of states. So here we can see Rhode Island is in this purple color, but you can see that in many other states, this is really rapidly rising. So my group has been really interested in trying to understand, as we bring these oysters back, what will happen, right? So they're powerful ecosystem engineers, they're filtering large amounts of water. So what will happen? And what I'm particularly interested in is this microbial process called denitrification. And I'm interested in that because denitrification is a natural filtering process. It's done by microbes that are ubiquitous in the environment. In fact, you have them in your body, you have them in your teeth plaque. So it's kind of gross, but also kind of cool. So these microbes are going to take biologically reactive nitrogen, the nitrogen that can stimulate phytoplankton or macroalgae blooms, and turn it into dinitrogen gas, or N2 gas. And when they do that, they are essentially taking it out of the system and putting it into the atmosphere and removing it from being able to be easily used by biology. So my group has approached this question in a variety of ways. We've done in situ incubations where we've tried to measure the process of denitrification in the system itself, as well as bringing some of the system back into the lab through and looking at these processes in sediment core incubations or incubating oysters alone. And today I'm going to focus on the sediments, the in situ incubations, and in the sediments. So just to make sure we have a couple of terms, I just want to show you what I'm talking about. So generally, right, what happens is we're going to take a large amount of organic matter. This is my examples of phytoplankton. These are some beautiful diatoms. They're going to fall to the bottom of the sediment and they're going to decompose. And then that decomposition process is going to use up oxygen in the surface sediment layers and spew off other nutrients like nitrogen, nitrate, ammonium, phosphorus, etc. And in that, when you add this organic matter below in the anaerobic or anoxic area of the sediment, you stimulate that process of denitrification, where this usable form of nitrogen is consumed and you turn it into N2 gas, which goes off into the water column. Now, this is one of these squidgy terms that scientists came up with long before I was around. And we call these are reactive forms of nitrogen, things like nitrate and ammonium, and we call dinitrogen gas or N2 gas unreactive. And I say that's a squidgy term because, in fact, there are some organisms that can use it, and those are called nitrogen fixing organisms or diazotrophs. And they take this N2 gas and turn it into a biologically usable form. So moving forward, we're going to be looking at these and focusing on these N2 fluxes, and I will remind you, but just remember when you see a flux coming out of the sediment, so a production of N2, that means we're measuring denitrification. And if you see a negative flux of N2, that is indicating nitrogen fixation or a consumption of N2. Okay, so some of the first work we did on this, we did in collaboration with Susie Avasian and Austin Humphreys, who is now tenured at URI. He was a postdoc with Susie at the time, and Bose Hancock at TNC. And we created these chambers that we could put in these different oyster habitats. So you can see this here, they have these rings, and we could place them either in bare sediments, not exposed to the oysters, or even underneath a rack and bag system, or even within a restored reef. And then when we went to go measure them, you can see it was a pretty intense effort. I'm happy to answer questions about that later, but we could put these chambers on it and essentially make this closed system to be able to measure the flux of gases or nutrients across the sediment water interface. And what we found was probably not that surprising, right? Since oysters are these really strong filter feeders, they're taking that organic matter, that phytoplankton out of the water column, they're putting it into their biomass, and then they're making feces and pseudofeces and dumping that onto the sediments, and that is going to stimulate microbial processes, including denitrification in the sediment. And so you can look here, just focus, we have bare sediment, culch, aquaculture, and reef in spring, summer, and fall. And you can see that regardless of the season, aquaculture, as well as the restored reef, had higher rates of this nitrogen removal or denitrification. And if we sort of put those all together for an average, that's even, you can see that even stronger here, where aquaculture and reef habitats really stimulate this process of denitrification. And it's not totally visible in this figure, but culch actually also does a really good job too, compared to bare sediment. I'm happy to kind of pontificate on why that might be later. Okay. So then this is my PhD student who's now a postdoc at Cornell, Nick Ray, and we sort of went back through that was done in the ponds. This work, we went into Narragansett Bay to look at the role of oysters. And there's a lot going on here. So I just actually really want you to focus up here on this figure. These are just a whole range of other nutrients, but this is the key figure, I think. Because this is this process of either denitrification here or fixation here. What's really neat about this is that we see that the sediments under oyster habitats change through the season. In the spring, they are removing nitrogen. In the summer, they're kind of doing both. It's almost like a net zero when you average it out. And yet in the fall, they're fixing nitrogen. And in fact, they remove as much nitrogen in the spring through denitrification as they add in the fall through nitrogen fixation. That's actually something that we see pretty similar to the subtidal sediments, so deeper sediments in Narragansett Bay. And then we also saw some small predictive patterns with things like phaeophyton, which is a measure of the dead, you can think of like a measure of the dead phytoplankton in the sediment. So it's kind of a proxy for food quality. Okay. Another thing that I was particularly interested in was whether or not the age of the farm matters. So these oyster farms go in, right? And we can imagine potentially like over time, as the farm is sitting there, that the system is changing in response to that, because you're now putting these oysters back in. They're going to be there for potentially many years, and they're sort of filtering all this phytoplankton and putting it down into the sediment, the organic matter in the sediment. And so I had some hypotheses that potentially like over the age of the farm, as the farm got older and older, maybe we would see a change in how much nitrogen is removed. Maybe it would just like linearly increase. Maybe it would sort of plateau at some point. And then maybe denitrification would become limiting or inhibited by some other processes or other environmental parameters as the farm increased. And I really liked this idea and this model, and I was so excited to test these hypotheses. And since Chris has been really honest about his models, I can say that I was, totally wrong, and that we didn't see any pattern in that. So we tested this farm over time, and you can see this down here is the age of the farm, from 0 being no farm, so control, all the way out to 7 years. And while there are some wobbles back and forth, there's no consistent pattern. One thing that is interesting is that when we just group everything together and compare bare sediment control to sediments under oyster aquaculture, we see that there's a strong stimulation of denitrification or nitrogen removal because of oyster aquaculture, but none of those beautiful patterns that I had hypothesized. And we do see nitrogen fixation throughout the time, and in fact, it dominates the control sediments. Okay. So then another thing that is interesting is this idea of oysters being a food source that potentially can provide protein with a lower sort of environmental impact. And one way to look at that is to look at the greenhouse gas footprint of oyster aquaculture. Before this work, this had not been done for oysters. It had been done modeled. People had created models for this, but no one had yet actually measured the fluxes of greenhouse gases in oyster habitats. And so we were excited to do that. And again, it was the same study where we were measuring it over time. And you can see that it's really a similar pattern. So nitrous oxide, methane, and CO2 don't actually show a significant change over time. In fact, there's no significant change even when you group all the time together to look between control and aquaculture. This matters, of course, because nitrous oxide and methane and CO2 are all powerful greenhouse gases, but methane and nitrous oxide are extremely powerful. So methane, for example, is about 25 times more powerful than carbon dioxide as a greenhouse gas, and nitrous oxide is almost 300 times as powerful. So small changes in the abundance of these greenhouse gases in the atmosphere can cause a significant change in our climate. And here we're showing that oyster aquaculture is sort of one way where you can get this protein source without having this large greenhouse gas footprint. And so for fun, we put this into context of some of the other protein sources that we commonly use. And really the take-home message is that you can eat a whole bunch of oysters and they may put a little bit of nitrous oxide in the environment, but it's negligible. And compared to chicken, pigs, sheep, or cows, oysters are really low greenhouse gas footprint. That was only done on one pond. So then we went to a series of other ponds, other areas in Narragansett Bay and in Duxbury Bay in Massachusetts. And we looked again just to try to get more seasonal variability because that was really focused in the summer, that previous work, to get more seasonal variability and also spatial variability to see how oysters could be changing the greenhouse gas footprint. And again, we see variability over the seasons, but when you're comparing between bare, so not exposed to oysters, and sediments exposed to oysters, you can see that there's really no change. Maybe there is like a slightly moderate change here for CO2 flux, but that does not equate to much. And even if we try to scale this number up, it doesn't really change our past estimates. So when you eat oysters, feel good that you're not contributing to your greenhouse gas footprint. Okay, so a couple more things I want to follow up on. One is that, we are one lab doing this work, and there's many other labs doing this work. And when you begin to look at the literature and try to understand sort of what is the consensus, do oysters, especially oyster aquaculture, does that increase nitrogen removal through denitrification? And it turns out when you look at the literature, it's sort of a mess. And so there are these different conclusions that say, like, we found no evidence to support that oysters increase denitrification, or our paper demonstrates that they do increase denitrification, right? So it's pretty wobbly. And there's lots of reasons for this, right? So you can imagine that people are using different techniques to actually measure the process of denitrification. They may be doing this different sized incubation chambers. They could be doing it in the field, so in situ or back in the lab. They could be including oysters in their work or not including oysters, just doing sediment underneath oysters. Sometimes they're looking at reef aquaculture. The site conditions can matter. So there's like a whole range of things. So Nick and I wanted to kind of attack that in a more quantitative way. So we undertook a meta-analysis approach where we went through the literature searching for keywords, and we found a series of papers that kind of fit the criteria that were necessary, which were things like directly reported rates of denitrification. We wanted some other environmental parameters, some real basic things, right, like temperature or salinity, knowing where the habitat was and that kind of thing. Our search started out with over 1,000 records, and we narrowed it down to 45. That's a very common thing in meta-analyses because of the way people report data. I just want to walk you through some of the key things that we found here. So these are showing all of the studies that were included, where we're looking at ammonia, nitrate or nitrite, and phosphate. Over here is telling you what the symbols match up to this figure. So if it's a reef system, it's a square. If it's a mesocosm, it's a triangle. And if it's aquaculture, it's a circle. And then we calculated this effect size G. And the key take home just to remember is like a small effect is 0.2 or less. A medium effect is 0.2 to 0.5, and then strong effect is over 0.5. And what I just really want you to notice is that, for example, oysters significantly increase the amount of ammonium cycling in the system. And they also significantly increase the phosphate flux. And these are both substantial effects, right? Greater than 0.5. And the NOx flux, the data is a little bit more limited, but what you can see is that the effect size is not significant. So right now we don't have the data to say whether or not they increase the flux of nitrate or nitrite, but they do significantly increase ammonium and phosphorus cycling. And when we look at denitrification, we can see that oyster habitats have a significant positive impact on the amount of nitrogen that's removed through denitrification. It's a strong effect, almost 0.7, and significant. And what's really interesting too is that we could find no difference between reef or aquaculture systems. So that implies that at this point, with the data we have in hand, oyster aquaculture and reefs are behaving similar in terms of the denitrification or the nitrogen removal process. And we also took a look at the greenhouse gas data that was available. And again, it's really hard to sort of pull anything out from this because there's not that many studies, but the studies that are out there, including the one of them I just talked about now, and then the other paper we just recently published, so it's not in here, but it looks like oysters don't impact the greenhouse gas budgets, which fits some of the same measurements we did. Okay, so is there hope for the future in terms of water quality and coastal systems and oysters? And I'm going to say resoundingly yes. So when we look at the amount of meat that we eat globally, you can see that the US, along with some other countries, eat way too much meat. And I think that if we could figure out a way to lower that footprint and replace some of that sort of land-based meat protein with protein from shellfish aquaculture, that could be a potentially really strong way to lower our environmental footprint. This fits with the long-term sustainable goals that the UN has put forth, right, with the goal 14 to conserve and sustainably use the ocean, seas, and marine resources. One of the reasons why oyster aquaculture I'm a big fan of it is because you're not adding anything to the system. So you put the oysters in, but you don't need to add food to them, right? They're going to get their food because they just sit there, and as the water flows by them, it's like a buffet that comes to them, and they can eat what's in there. So there are other types of aquaculture that can have a larger environmental footprint than oysters can, or other shellfish. Finally, I want to bring us back to this figure 2, these intense tables, and just show that while most of these systems had this dramatic reduction, right, in the ability of oysters to filter the water, you can see that there was this one system, Apalachicola Bay, that had this huge increase, and that's because of really focused restoration efforts. So when we put our mind to it, if we want to do something, we can in fact turn back time and help improve ecosystems. Okay. 2 more slides or 3 more slides, and then I'll be done. Motivated by sort of all of this and sort of the interest and excitement of this work, I hosted a workshop here at BU with collaborators from the EPA, Susie Avasian, and Bose Hancock and Skid Row. And we brought together over 30 people to work on trying to synthesize our understanding of how oysters can impact sort of nitrogen cycling and nitrogen removal. This is not everyone that was there, but it's most people. And what I'm particularly proud of is that we were able to bring in not just scientists, not just academic scientists, but we had state and federal scientists, we had undergraduates and postdocs, and we had oyster farmers. So we had oyster farmers at the table to come and discuss sort of what they needed to know, what was going to be helpful for them, and how we can start to build capacity for nitrogen credit programs and giving oyster farmers a potentially another revenue string, which could be essentially getting them money for the fact that their oysters are doing this ecosystem service of removing nitrogen. So we had 3 deliverables that came out of that 2-day workshop, and they were all published this year, which feels really amazing, especially after COVID. But the first one was this effort in trying to figure out how we can collect data in the most useful way to best inform not only the science, but also management and policy. And one of the things we really struggled with that meta-analysis was we could get the rates, but we had no ability to make any forecasts or prediction. And that's because the way the data are collected and presented and the types of data ancillary data, environmental parameters, for example, that might be useful, are not always collected or reported. And so this paper that just came out in L&O Methods is really providing a roadmap for how to move forward when we do this work in the future so that we can collect the best data to help inform management and policy decisions. And then hopefully build models too to be able to better predict these rates. The other paper that we had come out in Estuaries and Coasts is really looking at how sort of the opportunities and challenges of including oyster-mediated denitrification in nitrogen management plans, discussing, oysters are not going to be the end-all be-all. They can't solve all of our nitrogen problems, but they are one critical sort of tool in our toolbox that we should be using. And then the last paper that just came out like a month ago is this in Environmental Science and Technology that talks about sort of just the general idea of how oyster-mediated denitrification can be used in nutrient management. And it's written with the idea of people to the audience who may not always think of denitrification or be interested in it. And the idea is to try to put it into a broader context so more people can learn about it. The first two are open access, and this one will be open access in a year. But if you want any of the papers, you just email me. I'm happy to share them. Okay, so with that, I'm going to stop. I have thousands of people to thank, and especially thanks to Rhode Island Sea Grant, who funded the majority of this work. And I'm happy to answer any questions. Thank you.
Great, thank you very much for these presentations. So we saw some familiar faces in some of those pictures as well, so that was great. Let me start with a relative, we'll pose some of these questions. I know we're kind of running low on time. We started a little late today, so I apologize for that, but we'll start with I think a relatively quick question, and then I'll go back up to some of the earlier questions. But there's a question from the audience asking, how is this data being shared with CRMC, and is it impacting expansion of aquaculture leases?
I think that's for maybe, I'll take that one. I don't know if it's impacting aquaculture leases. All of the data we publish is open access, so anyone can have access to it. And we try to do our papers open access if we can afford it. And if we can't, just email me and I'll send them to you. So I hope it does.
Yes, and there's certainly CRMC folks that are listening in today as well. So, so that's another way that they're hearing about it. So great. Yep. Yes.
I would just add to that. So one of the things that I'm hoping to do, and been talking to various people about it, is to produce a new program at URI, one of these accelerated bachelor's to master's programs. And the idea would be that we would provide sort of modeling and data services to agencies like CRMC. And so as a precursor for this meeting, I'm teaching a class on the ROMS model right now. And so one of the students' capstones was to try to prepare some particle tracking in and around aquaculture leased farms for this meeting tonight. And of course, it's a student project, so it kind of went off the rails. But that's another way that I think that we can maybe try to get these technologies accessible to more people.
Great. I think some of the questions might have been answered during your talks, but there was an earlier question that was asking about let me read the question. It says, can you briefly explain the New Zealand Rhode Island connection? Is there similar topography, ocean water similarities? And this comment mentions that New Zealand studies are often cited in Rhode Island aquaculture papers. Can either of you comment on that at this point?
So I can say that we'll cite any aquaculture oyster paper we can find, and they're very limited outside of the East Coast. So there are some, of course, but they're pretty limited. So I would guess that New Zealand has a pretty large aquaculture programs, including integrated multi-trophic aquaculture. So that's likely why we cite them, because they're doing work that's similar to what we're interested in.
Great. We'll go down the list. There is a question here, but I think it was already answered. I'll ask you, and if you feel that you've already answered it, that's fine. So one of our participants asks, can you tell me about the March 2019 two-year study with Sea Grant about the statistical increase in denitrification due to aquaculture? Was there a statistical increase?
Okay, so I don't remember the years of the grants. So, but I could just say like in the salt ponds, we see a statistical increase. In Narragansett Bay, it changes seasonally. Overall, when you look at the total picture of all the literature that's available to directly measure denitrification, there is a statistical increase.
Great. I know it's 5 o'clock. I think we have maybe 1 or 2 more questions, and hopefully we can just answer those before we sign off. We know some people do have to sign off though right now. Let's see. So we have another question that asks, how long would it take to make a significant difference in the water quality of the bay with an annual coastal seeding?
That's a great question that I don't know the answer to. I mean, we're seeing seasonal changes, like each season there's a change. So I think it would be fast. Market size oysters are pretty bigger than just seeding. So definitely multiple years.
Great. Let's see. There's also a comment on Chris's comment saying, I love Chris's idea to create a framework model that CRMC and aquaculture applicants can use to look at these harder to quantify benefits and perhaps costs when evaluating an individual proposal. And let's see, I think we'll have the last question here for our presenters. So many coastal towns are doing resilience planning for erosion and sea level rise. Are there any artificial reef programs with the use of oyster beds?
Yeah, there's some work on that, not in Rhode Island that I know of, but maybe there is, but most of that work I know is down in Louisiana, where they've created oyster beds, and in North Carolina to look at storm surge mitigation. But that's a fun idea. If anyone wants to do that, let me know. We'll measure the nitrogen.
Great. All right. Well, I think with that, we will end our presentation portion, unless Chris, go ahead.
Well, I just wanted to, Wally, I ended my talk 1 slide too soon. So I need to, Wally reminded me that there's this data set that I've been collecting on physics has been going for years and years and years. So there's many people that have funded it, Rhode Island Sea Grant, Narragansett Bay Commission, EPSCoR, Vedelson. So, and also collaborators like Wally did a nice job of saying that everything she's talking about is a we, right? So on a huge group of students, and this work wouldn't have been possible without them.
Great, thank you for adding that, and thanks to you and all of your research teams and everything for doing this work and for sharing with us today. And I'm going to turn it back over to Azure for some closing remarks.
I'm sorry, can I ask a quick question? I'm on a phone and can't type anything in.
Okay.
I was just wondering, I don't totally understand how oysters can't be net carbon removers, just because their shell is primarily made of carbon, and obviously they're respiring and releasing CO2. But I would assume that over the years they're pulling out more carbon than they are adding, just from pulling zooplankton and phytoplankton out of the water column. Can you explain that for me?
Yeah, so Mike, that's a really fun question. And just briefly, it's pulling back on some chemistry that I haven't thought about for a little bit, but basically it has to do with the actual formation of the calcium carbonate. I believe CO2 is actually somehow released in that process. And so there's big debate over whether or not that is a substantial portion. So we don't look at that at all. That's not like, we're not looking at the calcification of the shell in that process. But what I can say is that what we have looked at, the CO2 is like slightly higher, just because they increase the amount of organic CO2 production in the sediments is slightly higher, because they're increasing the amount of organic matter that's feeding the sediment microbes. And so that increases respiration, which produces CO2. But there is a ton more work to do on that. It's actually a pretty sticky and hot topic to try to figure out how much carbon they're potentially sequestering.
Gotcha. Thank you so much. Great presentation.
Is there anyone else on the phone that is having a hard time putting in the chat? Feel free to chime in with a question. If not, I will thank Tracey for facilitating, and most certainly Chris and Wally for your time this evening and your great information. It's really been a pleasure, and thanks for all the work you have done and continue to do in this field. And also just look out for this presentation recording, and presentations will be posted on the Basant website. So you can get those there and share them with friends who might not have been able to make it. And I just wish you all a safe and happy holiday, and hope to see you at the December webinar presentation about shellfish restoration and enhancement with the town of Warren, with some experts in that. So look out for that invitation, and we wish you all well. Thank you so much.